Hi Year 13. This lesson we are going to move on with our NMR studies. We're going to turn to page 5 in your notes, and what we need to be able to do by the end of the lesson is we need to be able to assign chemical shifts to groups of protons. But before we go any further, we'll just recap where we left last lesson. Now you can see on the screen there we've got the low resolution NMR spectrum for ethanol. And if you remember back to last lesson, we said that there were 3 groups of hydrogens, and you can see that they're coded yellow, green and blue. We said that each peak is caused by a group of these equivalent hydrogens, and we said that the peak with the greatest chemical shift is the peak right beside or closest to the electronegative oxygen. So I can assign this peak as being caused by these blue hydrogens. I can assign the peak in the middle, close to the electronegative oxygen again, as being caused by these green hydrogens. And then the peak with the chemical shift of 1, I can say that these 3 yellow hydrogens are responsible for this peak. Now we're going to take it a step further this lesson. We're not going to forget what we already know, but we're going to try to assign chemical shifts in parts per million to each one of these groups of hydrogens. And we'll look at a few examples of that. Now if you look at the bottom of page 5, we have got a high resolution spectrum for 1-chloropropane. And a good starting point will be to draw that structure of 1-chloropropane in beside the spectrum. Now straight away you'll notice we've got our TMS peak, as you would expect, at 0. Remember, that's the standard. And then we have got, if you like, 3 groups of peaks. So I've got a peak here, it's been split up into 3. I've got a peak here split up into more than 4, and I've got another peak here split up into 3. Now I've got 3 main peaks. We will talk in another lesson about why these peaks are split, but before we leave that, we'll say that these peaks are split, and it's to do with the number of hydrogens on the carbon beside the carbon that we are talking about, but that's for another lesson. Now if I say that we have got 3 main peaks, and you can see here that we have got 3 groups of equivalent hydrogens. So if I colour code them, I've got some green hydrogens, I've got some blue hydrogens, and I have got 3 red hydrogens here on the end. So 3 groups of equivalent hydrogens are causing 3 main peaks on our spectrum. Now I can see that we've got a chlorine here. Chlorine's clearly really electronegative, so therefore it's going to de-shield these hydrogens, and that means that they will be shifted downstream, i.e. they will have a peak that has the greatest chemical shift. So the peak here with the greatest chemical shift is caused by these green hydrogens, and there are 2 of them. And we'll just keep our colour coding going. So they're caused by these 2 green hydrogens. Then the peak next to that is the peak with the second greatest chemical shift, and it's caused by these 2 blue hydrogens. Then the peak caused by the 3 hydrogens, they're furthest away from this electronegative chlorine, so they have the lowest chemical shift. This peak is caused by these 3 hydrogens. Now what we didn't say last lesson is that the area under the peak tells us how many hydrogens are responsible for that peak. Now you will notice that we have got red squiggly lines, and we have got 3 numbers that I'm circling now in black. Now these red squiggly lines and these 3 numbers are called the integration trace, and the integration trace can be used to give us information about the total area under the peak, and like I said, the number of hydrogens causing the peak. Now if I look at 1.2, 1.2 and 1.8, I want to simplify this ratio, and I want to divide across each of the 3 numbers to make that simpler. Now I've gone on to the wrong slide. If I divide across by 0.6, that leaves me with whole numbers 2, 2 and 3. Now I said to you that that gives you information about the area under the peak. And if you remember, we said that this peak was caused by 2 hydrogens, hence 2. We said this peak was caused by 2 hydrogens, hence 2. And we said that this peak was caused by 3 hydrogens. So this integration trace, when I simplify it to its simplest ratio, gives us the area under the peak, and it also gives us information then about the number of hydrogens causing each peak. And I know exactly the hydrogens, like we said at the start, because I know a little bit already about the chemical shift. Now, before we go on and we deal with chemical, that if I divide across, we're going to make sure that we've understood the number of groups of equivalent hydrogens in different molecules. Now we're going to take 3 examples, and it would be a good idea perhaps if you pause the video and try to answer the questions at the bottom. So if we look at methoxyethane, we're asked at the bottom of the screen how many peaks should appear in the proton NMR spectrum of methoxyethane. Now we're looking for groups of equivalent hydrogens, and perhaps if you pause now, draw out the structure on a piece of rough paper and try to answer that question before I go ahead and answer it. So I'm looking there at the molecule. I can see I've got a group of equivalent hydrogens here. I've got another group here, and I've got a third group here. So therefore, in an NMR spectrum for this compound, I would expect 3 peaks. Now, I want information about the area under these 3 peaks. The integration trace simplified down, well, it's caused by 3 hydrogens, 2 hydrogens and 3 hydrogens. So the peaks will be in the ratio 3 to 2 to 3. Now if we look at another compound, let's look at propan 1-ol. Now perhaps pause like before, make sure that you can pick out the number of groups of equivalent hydrogens. I've got 1 hydrogen here. We've got these 4 hydrogens, and I've got a group of hydrogens here. So I'm expecting 4 peaks. We've got 4 groups of equivalent hydrogens. If I look at the number then, I expect them in the ratio 1 to 2 to 2 to 3. We'll look at a last molecule. Let's look at propan 2-ol. Now I'm looking for groups of equivalent hydrogens. And remember, what I mean exactly by equivalent hydrogens is they've got to be in the exact same chemical environment. They've got to be joined to exactly the same atoms. Now propan 2 is a wee bit different. I've got these 3 hydrogens, but if you look carefully, they are joined to the exact same thing as the second group of 3 hydrogens. So these 6 hydrogens are in fact equivalent. So they will cause 1 peak. Then I have got another hydrogen here joined to the O, and I've got my 4th hydrogen here. So for propan 2-ol, I'm expecting 3 peaks in the ratio 6 because of those 6 equivalent hydrogens 1 to 1. And remember, it's the area under the peaks that will give us this ratio. Now, moving on then, we're going to start thinking in a wee bit more detail about the position of the peaks. Now, you can see then in your notes on page 6, you've got a copy of the NMR data that you will have in the back of your periodic table in the exam. So there's no need for you to learn these chemical shifts, but you will need to use this data sheet to predict the chemical shifts of certain hydrogens dependent on the molecule or the compound that they are in. Now it's important to remember as well, the closer to an electronegative atom, the higher the chemical shift. Remember, these electronegative atoms de-shield the nucleus of the proton, and if they de-shield it, it means that the peak is shifted downstream, i.e. the chemical shift will be higher. Now if we look at the molecule on the screen, we have got a molecule in ester, we've got methyl ethanoate. Now we're picking out our groups of equivalent hydrogens. There are 3 of them and they're circled for you. But we're taking this knowledge a step further and we are going to predict their chemical shifts. Now if we look there at the end, we have got 3 equivalent hydrogens, but we've got a CH3 next to this ester group. Now that's not one of the ones that you need for your exam, but I can tell you that we would expect these 3 hydrogens to have a chemical shift between 3.7 and 4.1. But you'll notice in your data sheet on page 6 that esters aren't listed there, so you'll not be asked about protons attached to an ester group. Now if I go to the other end then, I have got 2 equivalent hydrogens, but they are next to a carbonyl group, a C double bond O. Now if we look at this carbonyl group, their chemical shift would be between 2.1 and 2.6. And if I go then to our data, I can see that between 2 and 3, that is a proton that's beside a C double bond O, like we would find in a ketone. Yours is listed as between 2 and 3. The chemical shift data here is just a wee bit more exact, but you would list what you get on the back of your periodic table. Now if we go to the last 3 protons, we've got 3 equivalent hydrogens next to R, it says on the screen, and that just means an alkyl group. Now these are furthest from the O, they're furthest from the electronegative oxygen. Their chemical shift is going to be the lowest, and you can see that it is. It's listed there between 0.7 and 1.2. If we go to our chemical shift data, it's the one right at the top, between 0.5 and 2 for the saturated alkane. Now this chemical shift is just a wee bit more exact, but in our exam we would list between the 0.5 and the 2 because this is a proton beside another alkyl group like you would find in a saturated alkane. Now we're going to get a wee bit more practice at that. Now it's a good idea on page 7 just to take yourself through the worked example, and it's summing up everything that we have talked about so far, but it's another chance just to get that cemented in your brain. Now if you read to page 8 then, we're going to look at each of these compounds in turn. Now for each compound, predict the number of peaks, their relative peak areas, and the chemical shifts in a proton NMR spectrum. So the first thing that we will do is we will look for equivalent hydrogens and we will list the number of peaks. Now it might be useful to draw the structure of that compound out until you get good at it. So I can clearly see here that there are 3 groups of equivalent hydrogens. I would be expecting 3 peaks. Now I'm asked for the relative peak areas. Remember that's to do with the number of hydrogens. So the peaks would be in the ratio, their area, would be in the ratio 3 to 2 to 1. And you would be given that information like you saw at the start of the lesson in an integration trace. Now we're asked for the chemical shifts, and that's where you'll need your data sheet. Now if we go to the ones right at the end, these 3 hydrogens here, you can see that they're joined to a carbon, which is joined to an alkyl group. Just like a saturated alkane, their chemical shift will be between 0.5 and 2 parts per million. Okay, the 2 hydrogens in the middle then, well if we look at them, those 2 hydrogens in the middle then are joined to a carbon, which is joined to an alkyl group on its left. But if you look to the other side of that, to the right hand side, they're right beside a carbonyl group. Now this is going to be right beside a carbonyl group like you would find in a ketone. So the chemical shift copied from our data sheet will be between 2.0 and 3.0 parts per million. Now if I look at the one at the end then, this one hydrogen here. Now this one hydrogen is attached then to this carbonyl group, and that's like we would find in an aldehyde. So on our data sheet then, our chemical shift will be between 9 and 10.0 parts per million. Now I'm going to work through each compound in turn, but it might be a good idea if you pause the video, work through it yourself, and then just check that you agree with me. Now for the next one, I'm going to look at these hydrogens. I can see that there are 3 and 3 in exactly the same chemical environment. They're chemically equivalent, so I will expect one peak. Now its area would be equivalent to 6 hydrogens. Now when I look at them, I can clearly see that it's a ketone, so I'm reading off my data sheet that the chemical shift will be between 2 and 3 parts per million. Now moving on down to the next molecule then, and again if you're struggling to pick out equivalent hydrogens, it's a good idea to draw out the structure. Now I can clearly see that there are 3 groups of equivalent hydrogens. I'm expecting in this molecule 3 peaks. I'm looking for the number then, so the area will be in the ratio 1 to 2 to 3. Now I've got to go and assign chemical shifts to each of these peaks. Each of these protons, or each of these peaks. So this proton at the end then is joined to a carbonyl group like you would find in an aldehyde. So the chemical shift will be between 9 and 10 parts per million. The chemical shift then for this peak caused by these 2 protons will be between 2 and 3 because it's as if they're in a ketone, they're right beside a carbonyl group. So between 2.0 and 3.0 parts per million. Now our last group of hydrogens then, well they're joined to a carbon which is beside an alkyl group like we would find in a saturated alkane. So their chemical shift then will be between 0.5 and 2 parts per million. And I'm hoping you're getting the hang of this by now. Now it's definitely a good idea to draw out this structure for D so that we can see it. So we've got a carbon joined to a hydrogen attached to 2 methyl groups which is attached to a carbonyl which is attached to a hydrogen. So I can clearly see that it's an aldehyde and I'm looking to see about groups of equivalent hydrogens. Now I can clearly see that these 3 hydrogens are joined to this exact same thing as these 3. So those 6 hydrogens are equivalent. So I'm going to expect in the spectrum for this molecule that there will be 3 peaks. And the 3 peaks will be in the ratio 6 to 1 to 1. Now I've got to assign the chemical shifts for each of these. Now if I look at these 6 equivalent hydrogens here, they're just like as if they're in an alkane. We're looking at what's directly beside them. So the chemical shift of this peak will be between 0.5 and 2 parts per million. Now this hydrogen in here on its own then, well it's beside a carbonyl group like you would find in a ketone. So this hydrogen's peak will have a chemical shift between 2 and 3 parts per million. Now the one right on the end then is the one that's beside the C double bond O, like you would find in an aldehyde. So its chemical shift will be between 9 and 10 parts per million. Now in your notes we've got one last molecule to consider, and it's quite an important one. I've got C6H5. If you think back to your last unit that we studied in A2 Unit 1, I'm hoping that you think that that C6H5 is a benzene ring, because that's where we would have came across that C6H5. Now if I were to draw out the structure then, remember we've got our delocalized electrons in the middle. Then by convention we normally put our ethyl group onto the second carbon. We normally give this position 1 in fact. Then remember, because it's C6H5, that there's one hydrogen joined on to each of these carbons. That one's not going to draw. Now we're looking for groups of equivalent hydrogens. So I'm hoping that you see that these 5 that I'm going to circle are all in the exact same chemical environment. They are chemically equivalent. So therefore I'm expecting 3 peaks, and I'm expecting these peaks to be in the ratio 5 to 2 to 3. Now I need to assign the chemical shift data then for these 3 peaks. Now these 5 carbons here that we would find on an arene or a benzene ring, and it's listed on the back of your data sheet then, will have, their peak will have the chemical shift between 6 and 8 parts per million. The 2 then, hydrogens that we come to next then, will be between 2 and 3. Now the last 3 hydrogens, it's just as if they're in a saturated alkane. They don't look as far as the benzene ring, they're just looking at what's directly beside them. So their chemical shift will be between 0.5 and 2 parts per million. Now if you look at the top of page 9, we have got 2 more little NMR spectrums that we're going to consider. For each proton NMR spectrum, identify the protons responsible for each labelled peak. Now you're given the molecule for each one. Now I'm just going to draw out this molecule so that I can clearly see what's attached to what. Now I can see that we have got 2 bromopropane. Now we're asked to identify the protons responsible for each peak. I am hoping that you see that these 3 protons are equivalent to these 3 protons. Okay, they're not directly attached to this electronegative bromine, so therefore these CH3s, these protons that are part of the 2 CH3s, will have the lowest chemical shift, and I can go and assign these CH3 protons to the peak B. Now that only leaves the proton then that's attached to the CBr. Now I'm going to apply a similar method then to the ester that's down below. I'm going to draw out the structure just so that I can clearly see what's attached to what. And you can see that we've got ethyl ethanoate, and I'm looking for groups of equivalent hydrogens, but there are none. There are 3 groups, all separate. I've got 3 peaks. I'm looking to see what's directly attached to the electronegative oxygen, and that's the CH2 group. So it's the CH2, I'm going to underline, that's attached to the CH3. Okay. The next group then is our CH3 that's attached to the ester group then. So it's these 3 protons causing peak D that are attached to this C double bond O. Now that leaves then the CH3s right at the end of the ethyl side chain. They're furthest away from the influence of these electronegative oxygens. So the final peak E is caused by these 3. Now, folks, before we leave this lesson, if we look then at this 1 bromopropane, and we'll sum up everything that we've covered. So in 1 bromopropane, there are 3 groups of equivalent hydrogens, and we are expecting 3 peaks, and you can see that we've got 3 peaks. Remember then that the area under each peak gives us information about the number of protons causing each peak. So we have got the area in the ratio 2 to 2 to 3. So this peak with the chemical shift around 1 has been caused by 3 hydrogens, and this is the peak that's furthest away from the electronegative bromine. Then we have got a peak roughly at 2 parts per million caused by 2 hydrogens, and that's the one that is next furthest away from this electronegative bromine. Then the peak between 3 and 4, the last one, is caused by the 2 protons that are attached to the carbon that's also attached to the bromine. Now if you look to the right then, we're thinking of the exact same molecule, but we're going from the low resolution spectrum to a high resolution spectrum. And you can see that these peaks are split, but you can also see that we've got a little trace there just to show you that those peaks are the same ones, but they have been split up. Now it's important to realise that they are split, and they are split according to the number of hydrogens that are on the adjacent carbon atoms. And I'm just going to cover this before we leave it, but we will come back to it in the next lesson. So this peak at roughly 1 part per million was caused by 3 hydrogens, and it was caused by these 3 hydrogens. Now if you look to the neighbouring carbon, there are 2 hydrogens on it, and it always splits up into 1 more peak than the number of hydrogens we've got on the neighbouring carbon. So it's caused by 3. There are 2 on the neighbouring carbon, so you can see that it's split into 1 more than 2. It's split into a triplet. So this splitting gives us information about the number of protons on the adjacent carbons. Now if we look at these 2 protons, okay, these 2 protons then are causing this peak. Now you can see that this is split up into many peaks. We would just call this a multiplet. Okay, now look at the protons on the adjacent carbons. We have got 3 to the left and 2 to the right. That makes a total of 5. So it's actually split up into 6 tiny peaks. It's called a hextet, but it's enough for us to remember that it's a multiplet. Now if we look then at the last 2 hydrogens, they are causing this peak with the greatest chemical shift, because remember they're attached to the carbon that's directly attached to the bromine. If we look at the adjacent carbon, well, there are 2 protons on that adjacent carbon. Remember it splits up into 1 more than that, so again this has split up into a triplet. Now we will come back to this next lesson, but there's just a wee bit of a preview of our next. Now if we sum it all up, an NMR spectrum therefore then tells us about the number of hydrogens, or the number of peaks, tells us the different types of equivalent hydrogen groups. The position of the peak tells us about the chemical environment of the hydrogens. And remember, we get that from the back of our data sheet. The area under the peak, and we would get that from the integration trace, tells us how many hydrogens we have causing that peak. Now, this splitting is sometimes called the spin-spin coupling pattern. That's basically the splitting pattern, tells us the number of hydrogens on adjacent carbons. Folks, we're going to leave it there for today, but you can look forward to the next lesson where we look at this splitting pattern in a bit more detail.